Genetics initially in the last century was really coming with the great information that now we have genes and genes develop, who we are. And then they realized that it cannot explain sufficiently the information, what's happening. They practically realized that genes are there, but they're not always active. So they were looking into solutions on how to explain that. For example, you have twins that have identical genetic information, and what happens is they become different. So how can you explain that if the genetic information is exactly the same when they were born? They came from one cell. So, and then, I don't know if you heard, but there is like a stream of the science that is called epigenetics that is actually working on this information. So, for example, epigenetics started realizing that some genes are activated and the others are not. So they went into findings on why some genes are activated and some are not. What are the biological causes? And they actually found biological explanation of why some genes are activated and some others are not. Without changing DNA, DNA stays the same. And then from DNA, one gene is on, one gene is off. The same gene is on, the same gene is off. So that can also explain a lot of things that we do in our therapeutic work when we make changes, because we can help that some genes are activated or disactivated for our work. We will talk about this a bit later.